Hello everyone, today we will learn about FASTQ file format. FASTQ file is the most common type of file format that stores raw sequencing information. That means this file has the nucleotide basis of a DNA sequence, as well as this file contains the quality scores of each of the nucleotide bases. Most of the sequencing machines provide raw sequencing information as FASTQ file. Also, most of the bioinformatics tool can recognize FASTQ file format as input for downstream analysis. FASTQ is a text file. The common file extensions are .fastq or .fq. FASTQ file could contain millions of sequences. Therefore, the file size could get much bigger. To reduce the file size, standard text compressions are frequently used. This is an example of a FASTQ file. For each entry in the FASTQ file, there are 4 lines. In other words, for each of the DNA sequence, there are 4 major sections in a FASTQ file. First one is the identifier that always starts with @ sign. The identifier is followed by the DNA sequence, and there is a separator using + sign, and final section is the quality score. Freight quality score or Q score is the single letter representation of the error rate for each of the bases. DNA sequencing machines do make mistakes during base calling. Therefore, those single letter quality scores represents the probability of incorrectly identified bases by a sequencing machine. I have made a separate video on freight quality score. Please check out my video. There is no standard definition for the identifier line in a FASTQ file. The identifier line could be different depending on the sequencing platform or the data sources. This is an example of Illumina's standard identifier line. Those number here represents the specific information of this sequence. For instance, the first string here represents the instrument ID, which is followed by run number, flow cell ID, lane, tile, X and Y coordinate information, read number, for example here is 1. Read number is 1 if it is a single end library, or in the paired end library this number could be 1 or 2. Then pass filter, if this sequence did not pass the filter then it is no or N, otherwise it's Y or yes. Control bits, also it has additional barcode or adapter information. I want to mention that often this identifier line could be very simple, that only contains the read ID and the number. There are many bioinformatics tools available to analyze FASTQ files. To check the quality of DNA sequencing bases in a library, we can use FastQC or MultiQC. For all downstream bioinformatic analysis, we remove the low quality nucleotide bases from the raw FASTQ files. For that, we can use cut-adapt or trim-o-matic. Often, we may need to convert the FASTQ file into FASTA file, or we need to convert the quality encoding between different sequencing machines. SecKit is a very useful tool for those tasks. Please check out my other courses on my website. Thank you for watching this video. See you in the next one.